Good afternoon, everyone. Bonjour à tous. The theme of this year's edition of the KIKK Festival is actually very close to me. I'm a biologist and a visual artist. And today I'm going to try and talk about hybrid projects between biology and art in quite a few different ways. These are the topics that I would love to address, illustrated by different topics. The first part is going to be about violence in nature. Also have a few projects that are dealing with engaging with nature as a co-designer, a co-creator. And then last but not least, I'll be talking about technological agency. As I said, I'm a biologist, and I think maybe some of you in the room, I'm sure they already know this story. My background is in freshwater biology, ecology and developmental biology. And I did years of research on this particular topic. I worked on deformities of the teeth of larvae of non-biting midges. Very specific topic, typically for science. You might wonder, why on earth would somebody be interested in the teeth of a larva of a non-biting midge? Well, the thing is, these larvae, they live in the sediment of streams and ponds and rivers. And when these ecosystems are contaminated, the toxicants end up in the mud, in the sediment. And because these animals, these larvae, live in such proximity with the sediment, they take up a lot of those toxicants, and their teeth, as a result, their teeth deform. So you can actually use those deformities as an indicator of environmental pollution. This is called biomonitoring. So it does make more sense if I explain it like this, of course. Now, the kind of work that I was doing was the following. I would go to a river, we would sample mud with a net, then we would go back to the lab, we would take out all these small little red bloodworms. We would have to kill them by throwing them into alcohol, like almost 100% alcohol, which kills them pretty much instantly. And then one by one, I would have to cut off the head of each larva with a micro scissors, cut off the head, then take the head and put it in a small cup, a porcelain cup, filled with acid on a hot plate, and I would boil the heads. And why would you boil heads of insects? Well, the thing is, insects have an exoskeleton, right? So the head is like, the skeleton of the head is on the outside. So if you want to look at the head under a microscope, you need to remove the tissue which is inside. That's why we were boiling them in acid. So all the tissue gets out. And then you take the little head, you put it on a glass to make a slide for a microscope observation, and then you kind of dissect the head with those same scissors and with pincers. It's all very tiny work, of course. A head like this is only 1 millimeter across. You can imagine the kind of skill that you develop after a few years. I did these things together with a few other technicians in the lab, and I looked at I don't even remember, probably 10,000 or something of these little head capsules to figure out what was going on. And this is the kind of research that we were doing. What you can see here is results of I was actually doing a PhD in this. And what you can see on the graph is actually the frequency of deformities and then different locations in a stream. And where the little asterisk is, that's where a factory was discharging its pollutants. So you can really see, as soon as a factory was discharging pollutants in the stream, the frequency of the deformities went up. That's just one example. Now, when I was doing all this kind of research, I've always been interested in art and science. And I kind of knew that I wasn't going to spend the rest of my life looking at those teeth, looking at the world through a microscope, and I was always interested in the arts. And that's parallel with this work. I also went to art school. I basically did a degree in photography, evening and weekend class. So I was combining these two practices. And after some time, I got really interested to develop myself full-time as an artist. I quit science, I finished the PhD, quit science, and moved into the field of art. And it took me some time, but after some time I got really interested to bring back biology into my daily practice, but as an artist, very differently. And one of the first works that I generated is this one that I'm going to talk about. I don't know how many people know about this particular organism. It's like I'm giving a biology class instead of an arts talk. This is a Daphnia. It's a water flea. These are very little tiny organisms that are living in fresh water. You might have seen them. They're like little specks, and they they basically have a movement where they jump up in the water twice and then they sink. Hop, hop, sink. Hop, hop, sink. That's how the scientists actually call their behavior. They're very interesting organisms, of course. I think probably all organisms are interesting. But they have two ways of reproducing themselves. They can reproduce asexually and they can reproduce sexually. So that's an interesting option they have there. And their favorite fish food. Fish love them because they're full of protein. Now, I made an artwork with this organism, and I did this together with a former colleague of mine, Luc de Meester. He's an evolutionary biologist at the University of Leuven, and we built an interactive installation with this organism. And one of the very interesting things about this organism is that it's very sensitive to peculiar colors of light. It has evolved this behavior. It is very much attracted to yellow light, and it is very much repelled by blue light. And there is an evolutionary reason for this. Yellow light indicates food particles in suspension. They're filter feeders, so whenever they see yellow light, they think they can filter out nutrients from the water. Blue light indicates visibility for predators, so that's what they try to avoid. And based on this behavior, we developed a Darwinian evolutionary artwork. And I'm going to explain this to you using this video. The sound of this video shouldn't be too loud. Let's see. All right. So what you can see here is the water fleas just swimming close to the surface of the water because on top of the water there is yellow light. And like I said, they're really attracted to this yellow light. You can also see multiple generations of them. You see smaller ones, bigger ones. Just like insects, they actually molt. Here you can see how they're nicely mathematically distributed under each patch of yellow light. It's pure math. You see the whole population is nicely distributed and clustering under each yellow patch of light. But down below, there were fish. And the fish were separated from the water fleas with a false bottom. There were holes in that false bottom. The fish could not swim up, but the water fleas could swim wherever they wanted. That was kind of the basic standby setup of the entire installation. We also did multiple replications of the same experiment. So you can see 5 replications, so we could actually run statistics after the experiment would have been completed. This is the lighting system, and the lighting system was actually developed together with Philips, the electronics manufacturer. And here you can see what happens if a spectator moved closer to the artwork. Basically, the presence of the spectator was picked up by a sensor, and blue lights were turned on. In the beginning, there was still, the yellow light was still on, but after some time, the yellow light was turned off, and the blue light got more and more intense. And what was happening was the following. Because of the blue light, the water fleas were afraid, they swam down, and they were eaten by the fish. So basically, people instigated this violent drama inside this artwork. Now, it's way more interesting than that. The thing is that their behavior is genetically coded. And there's variation. There's variation in genes, there's variation in behavior. So not every single water flea is equally afraid of blue light. So some of the water fleas would actually stay up because they were less afraid. They would survive and they would reproduce. They reproduce really fast and would transmit their genes to the next generation. And basically what we were trying to do here, and successfully did, was we created basically an unnatural behavior. We basically created animals that were attracted to blue light instead of running away from blue light. Animals that in real nature would be wiped out straight away, but in this art context were thriving. And after which we actually tested this behavior, and as you can see here, it was basically a little experiment. We took the water fleas and we illuminated the water column that you can see on the left from the top, and then we could see how they distributed themselves. And so if they were afraid, they would go down. If they were not so much afraid, they would stay up. And then based on that, we could prove that the water fleas that were exposed to that specific experiment that I just said were less afraid of blue light. Now, like I said, these were all pretty violent interactions between man and animals. I've also been exploring other ways in which nature gets its agency, gets its own agency, but in a kind of different kind of relationship. This was actually the very first work that I made in which I combined my background as a biologist into art. What you can see here is basically a canvas. I installed a system in which water was circulating over the surface of the canvas, was then captured in a small container at the bottom, and was recirculated over and over again. Subsequently, after installing this, I made a walk around my studio and I collected objects that were outside covered with a green layer of algae. And I got back into my studio and I infected the canvas with these objects. And then I stood back and I waited. And what you could see is that over time, an abstract pattern started developing itself over on the surface of the canvas. So you got basically an organic, breathing, abstract painting. Like I said, it's an early work. It's the first one I did. It's quite simple in its approach, but it really resonated with me very strong. And there is some interesting relationship here. You basically, as an artist, you create some framework, you add a living organism, and then the organism starts doing its own thing. So there is this unpredictability, which I found very fascinating and very interesting, and an agency that we as humans perceive as creative or aesthetic, which is obviously not really the intention of the algae, because they don't really care whether they're on a painting or on a brick or on some other object. Now, of course, you can also, and I got really interested in this, like I said, and this is a project that I did, which is basically based on the history of film. But the same interest in using organisms' agency and using organisms' processes to create very dynamic and somewhat unpredictable art, where a different relationship emerges between the artist and the artwork. And it's inspired by these kind of famous examples of experimental cinema. This is the Free Radicals experimental movie by Len Lai, and this one as well, Stan Brakhage, Mothlight. Very famous examples. But what they were interested in was, they were not so much interested in the illusion that is generated by cinema, which regular cinema, mainstream cinema is mostly working with. They were actually interested in questioning the medium itself. It's a hypermedial interest. Instead of aiming for transparency of a medium, transparency of the film medium, where you forget the medium, you forget you're in a cinema room and you're really absorbed by the medium, they were stressing from a hypermedial perspective, the nature of celluloid, the nature of the film strip. They were scratching in it, adding elements to it, and then working with that. And so I was thinking about, how can we translate this to contemporary times? How can we translate this to what we're working with today? Digital media. There used to be, it's much less now, but like 10, 15 years ago, there was a lot of talk when people were talking about digital media and media art, about the immaterial nature of digital media. I was always surprised how people were stressing this. And as a computer builder, I love to build computer systems, and it's even one of my projects. I was always surprised. I was like, no, there is a lot of physicality to digital media. And it's actually really interesting. So why do these people get this wrong? So maybe this is interesting to work with. So I really wanted to make an investigation into the physicality of digital media, inspired by that tradition from the history of film. And so basically the question that I developed was, can we manipulate digital media just like those other artists did using biology? Can the growth of organic life on digital media cause visual glitches? This was kind of the interest in the project. And this is the kind of experiments that I started doing. Growing molds on hard drives with data and then trying desperately to capture the data from the hard drives, which obviously failed over and over again. I had exhibitions with this. There were piles of hard drives that I just did all kinds of physical experiments with, and it never worked. And then my friends from Fluxspace made an interesting short video about this, in which they talk about how they looked at me trying to work with these hard drives. Now, I didn't give up, of course, and I really still kept on searching for ways to make those two worlds meet, the physicality of hard drives and biological organisms, and ended up working with insects, back to insects. What you see here is a Madagascar hissing cockroach. It's usually used as food for pet snakes and pet reptiles. And developed the idea with some other people to build armor for insects, and armor that would contain electronics that would actually disrupt and destroy data. That was the idea. So the first step was, okay, how are we going to build the armor? And I walked up to a jewelry designer, because they know how to build really small things, very intricate things. And Walter Blesleers, he's a jewelry designer from the city of Antwerp, where there's a lot of jewelry designers. And so we started prototyping these armor pieces for hissing cockroaches, as you can see here. And then in a later stage, we actually developed silver pieces that could actually be fitted onto a cockroach. And so the idea was actually that through picking up electromagnetic signals from computer components, that these signals would actually disrupt the data on the same instruments. And yes, I have a little overview here. So the idea is this magnetic induction. If you have a spool of copper wire and you keep it somewhere close to a digital device, it will pick up the electromagnetic field, it will generate a current, and that current can then be transformed again in a different location, once again into an electric field, and that can disrupt data. So that was kind of the idea that we had, that the computers, through working, were generating their own magnetic fields, and then insects would play around with those fields and would actually start destroying some of those data. And we built several installations around this. This was in Beaux-Arts in 2010, where we had the whole equipment set up, and it was like a small kind of test bench where we could explore this idea. We also made, together with Shelbatra Yashari, she's a filmmaker based in Brussels, we made a sort of experimental film in which we kind of documented the process of trying to disrupt these data. And I'm going to show you a short clip from this movie now. There's of course also a different way in working with organisms, and instead of just inviting them to contribute to some aesthetic process, some visual process, you can actually also tap into the intelligence of organisms, of course. And what we see here is slime mold. I'm not sure how many of you are familiar with slime mold. It's a very interesting organism. It's been used a lot recently by designers because it has some particularly interesting characteristics. Some of the species of slime mold, it is kind of hard to distinguish its identity. The thing is that in slime mold, it has two different stages. On one hand, in this particular species, it lives at the individual cells live independently as individual organisms. And when it's time to reproduce, they all come together and they form one organism, which is a little weird to think about, but they form this one singular organism that then reproduces itself, and then you have all these individual cells again. So that's kind of the weird world of slime mold. Now, slime mold is also famous, like I said, also, for example, also in the design world, because it has a sort of intelligence, which is quite astounding. For example, if you put it in a maze, and you put an attractor at the outside of the maze, so you put the slime mold at the center, you put an attractor like an oat flake at the outside of the maze, it will actually find its way immediately outside the maze to get to the flake. So there is some interesting potential here, some interesting computational potential here. And this is something that we explored in a project that I was part of a couple of years ago, together with researchers at the Angewandte in Vienna. It was called GRAB, Growing as Building, a project specifically looking at living architecture and the possibilities of creating architecture that would evolve and grow over time. And so we did a case study on this particular site, which is the Mounsell Fort. It's a particular interesting location in the UK. It's like a World War II building, a World War II infrastructure, kind of defense infrastructure. And it's just sitting there, but actually there's quite a bit of possibilities that could be done there, could be repurposed in all kinds of ways. But those different units that you could see, those different architectural units, they could be connected in interesting ways, differently than originally intended, and that's how we used the slime mold. We basically translated the layout, the architectural layout of the site into a 3D grid that we 3D printed, and then we indicated the important locations of the architecture using those oat flakes. And then the slime mold would be connecting those in a three-dimensional way, and then we could scan the three-dimensional structure that arises, and then you could use that within your architecture. So basically, you could, in this way, using a 3D printed grid, using the intelligence of slime mold to connect structures in 3D space, and then scanning that, you could actually use that as an instrument in architecture. So that's the kind of interesting, a different paradigm of working with organisms within art and design, which I find is actually really interesting. Now, what I'm going to talk about next is more about looking at technology as an agent. So far, I've been talking about biological organisms as agents, as non-human agents involved in all kinds of relationships with people, with man. We can, of course, also have to look at technology as another agent. So there's like biology, technology, humans, all interacting in different ways. And the field that I'm going to talk about now is called morphogenetic engineering. This is a relatively recent field in engineering. I find it tremendously interesting, specifically because I'm a developmental biologist. It basically positions a different paradigm in engineering, a different way of building systems and design. Instead of having a top-down approach, instead of having a blueprint that has been made by a few people and then has to be executed, in morphogenetic engineering, you're not so much concerned with making the detailed blueprint of the final design. You're actually working on individual small units, robots, for example, that as a swarm can build complex structures. And this is inspired by termites. It's one of the examples they're inspired by. As we all know, termites build termite mounds, but the thing is with those termite mounds, they're really well-engineered structures. The challenge for a termite mound is really how do you bring in fresh air and keep the hot air out? That seems like really difficult, and they solved it. They solved it by using a particular way of building ventilation shafts in a particular structure, and they actually managed this. Now, when termites are building a termite mound, you don't have one single termite that is running around with the master plan telling all the other termites what needs to be done. There is no star architect in the world of termites, of course. So how do they do this? And they do this because they use a bottom-up design approach. And the individual termites interact in particular ways with particular rules, and out of that emerges the structure. This is very interesting. So in morphogenetic engineering, you actually work on the individual units, the individual agents, their properties, and the rules on which they interact. And the advantages of using this kind of approach are first of all, this approach can result in solutions that were unexpected, because you're letting the system evolve and emerge its own solution. So it goes beyond what you as a human or with using technology could even come up with. So that's definitely a major advantage. It also results in massive parallelism, because there's all these agents that are working on the structure simultaneously. And there is no central bottlenecks, so things can go really fast. So there are all kinds of advantages in using this. I'm not saying that engineering should go all this way. All engineering should become morphogenetic engineering, but I'm just saying there's tons of potential there, and I think it's going to be part of our future. So the context in which I'm applying this is interstellar travel, and this is what I'm currently doing at Delft University of Technology. I'm working on research on developing concepts for spaceships that can travel to a different star, starships. Now, the central challenge in interstellar travel is the following. You're dealing with unpredictable futures, uncertain futures. In contrast with previous missions that have been planned or are being planned in the solar system, we have to use a different approach. For example, if we look at the Apollo moon program, of course, they didn't go to the moon in one shot or even in two shots. They went to the moon closer and closer, each time coming back to Earth, fixing and optimizing the system, and then going back. It's all about trying to understand and map all contingencies and all risks that are involved. Now, the thing is, with interstellar travel, you can't do this. You can't just go a little interstellar and come all the way back and then fix things on Earth. That would be completely very inefficient and a very long process. So the challenge is for engineers, they have to develop a starship for a series of challenges that they have no idea about. This seems like impossible, right? And then this is where evolution comes in. Could we develop spaceships that actually evolve over time and that actually adjust and adapt over time during their journey, so you don't have to worry about incorporating solutions for all things that could go wrong, because the system independently can already handle it. You're basically giving agency to the system to deal with those uncertain futures. I'm doing this with a team of volunteers, mostly from TU Delft, but also from outside university, both researchers and students, and we've been working on this for a little more than 2 years. So how would you make this idea operational? How would you make a spaceship that actually evolves? Well, we're using 3D printing. 3D printing is one of our core technologies in our concept. The spaceship is actually printing itself during its journey, and that's one way in which you can have a space infrastructure which is adaptable and evolvable. And if you're printing, you need resources. You're not going to bring all those printing resources from Earth. That would be a waste of energy and of money and of time. There are resources out there. Asteroids, they contain a lot of material, a lot of different types of materials. We're talking about metals, we're also talking about water, organic compounds. So they're a really good resource for building stuff in space. So the idea we developed is that we could mine and transform an asteroid over time and then use those materials to print a spaceship. The idea of an asteroid starship is actually nothing really new. The concept itself has been, I think, one of the earliest examples is from the 60s. Dandridge Cole, an engineer, came up with an idea of transforming asteroids into spaceships, but also in science fiction it's actually really present. Now, if you want to develop an architecture that is being printed over time in a very hostile space, such as deep space, it makes sense to use a modular architectural approach. You're not going to build a big central shape, because then it becomes very difficult to extend that shape. But with smaller modules, you can keep on adding modules, and you have a lot of flexibility, basically. So in our research, we basically defined different types of modules that have different functions, and that's how we compose our spaceship. And this is an artist's impression of how it could look like. The asteroid is gradually mined out on the inside, and in the space that is made available on the inside, the architecture is being developed, but also the architecture is also developed at the outside of the asteroid as a tail behind the asteroid. And in this way, the asteroid is not just a resource for the 3D printing, but it is also functioning as a shield for impacts of small particles during the journey. The way we're printing, the way it's being printed is by using mobile printers that are just like a bit comparable to the story of the termites that I just explained. They're printing at multiple locations at the same time with a high flexibility. And we're also using differential printing, which means that depending on the materials that are available, we're using a different material to print the architecture with. So it's really a mixed material approach. Our goal is to build a computer simulation in which we can test these ideas. This is not just speculative design, this is actually simulation research. So we're building a computer simulation in which we can simulate an entire journey, let's say a 100-year journey, through a predefined interstellar trajectory. Now, there are two main challenges when we're talking about interstellar space. One is what I just talked about, small particles that can cause a lot of damage because you're traveling with a very high speed through interstellar space. The second one is radiation, cosmic radiation. Here, also at the ISS, we're pretty well protected from cosmic radiation because of the Earth's magnetosphere. Once you're in deep space, this is not the case anymore. And cosmic radiation could wipe out all life on board of your spaceship. So that's why we came up with the second idea, not just a growing starship, not just an infrastructure, an architecture that is actually expanding over time, but an architecture that can reconfigure itself, that can shapeshift, that can take a different shape according to a new challenge that comes up. And this is specifically possible because we're using that modular architectural approach. We can really reshuffle those modules with different functions to different areas, and we can reconfigure the spaceship according to its needs. For example, if at a certain point high radiation is expected from a particular site, a lot of shielding could be moved over, and a lot of more sensitive modules, for example, containing life, could be moved to a different site. Now, this is a bit the summary. So the idea is basically that the spaceship can sense ahead. It has a sensing horizon, just like in robotics. It cannot gauge the entire journey, but it can gauge a good part of what's coming ahead. If it's very similar what's coming ahead, then the structure can keep on growing in the same fashion as it is. It's basically like crystalline growth. If what's coming up is very different, then it needs to evolve, and it will use genetic algorithms to optimize its own morphology in order to tackle those new challenges. This is the kind of experiments that we envision. We plan an interstellar journey. We make a scenario, we create a scenario with different particle densities during that 100-year journey, different radiation events, potentially multiple radiation events on top of each other. And then we start virtually a journey with a virtual spaceship connected to a virtual asteroid. And then we stand back. And we look how the architecture is adapting and evolving itself, and we study that. And then we can run a mission through exactly the same space, the same challenges, but using an asteroid with a different chemical composition, and start comparing and start learning. So that's basically what we're doing here. Now, we also make art installations with this work. We just finished basically showing a multimedia presentation, a multimedia installation for the Breda International Photo Festival in the Netherlands. And there we showed different types of images that were basically visual studies of the research that we're doing. So the project, which is called EAS, Evolving Asteroid Starships, is really deeply hybrid. It results at the same time in scientific papers that I'm presenting at scientific conferences, and it results in artworks and in art installations. I'm going to show you a video that is basically a part of what you could see at the Breda Photo Festival. think I have to speed up because we're running out of time. Now, so what I've been talking about is basically developing technology with its own agency by modeling it on biology, turning a spaceship almost into a living organism in its behavior. Now, it wouldn't make sense if by using this approach you come up with a beautiful strategy to create spaceships that actually arrive at destination structurally sound, all in one piece, fully operational, if everything inside, if all the wetware inside is dead. So second part of our simulation is actually simulating a regenerative ecosystem inside of our spaceship. And so within this, with this concept, we can also use the asteroid as a resource to grow and maintain an ecosystem. And as a model for the ecosystem, we're actually using an existing ecosystem, an existing space-oriented ecosystem from the European Space Agency, which is called MELISSA. And the MELISSA project is basically an artificial ecosystem that takes all components, all waste products from humans, everything, toilet waste, urine, CO2, sweat, breaks them down gradually in a series of bioreactors with different bacteria, and turns all of that into nutrients for plants that provide oxygen and food for the astronauts again. So it's a circular system. And so we're modeling that using a technique which is called agent-based modeling, which I'm not going to explain in detail here, just to mention, but it's quite related to computer games, actually. You have individual agents, virtual agents, plants, bacteria, or bioreactors, astronauts, all interacting, and then you can study under which circumstances the whole thing so they survive or not. And one of the things you have to do is you have to map all the mass flows, basically all the molecules that are being transmitted between all those different organisms. And this is chemistry. It's one of the things that we do. We do all the math of the chemistry. But there's an interesting consequence. If you build a system in which all these molecules are being transmitted, are being exchanged with each other in such a very precise way. First of all, imagine you're living in a closed space with a few bacteria and plants, and you're constantly exchanging all these molecules. At a certain point, the atoms that you're consisting of have been in the bodies of those plants and bacteria. And if you're with two astronauts, you're basically starting to share the same atoms. Your constitution starts to be shared with all the others. You basically create a meta-body in which everything is connected in a very deep physical way, which I find philosophically quite interesting. But also, you become hyper-dependent on each other. Every single molecule in such a system is valuable. It's a very different ethics than living in the open world here, because we have an atmosphere that is endless, pretty much. The oxygen is just flowing in from nobody knows who produces it. But in a system like this, you're almost aware of every single molecule of oxygen you're breathing, where it came from. And if one of those organisms dies, you're in deep trouble, because you really need to balance that whole system. So it really generates a complete different perspective of what it means to be alive, connected to different organisms. And I think with this, I'm closing my talk. Thank you so much.